Thank the organizers for inviting me. It's a very prestigious honor to be here with you today. As an academic person, you get to travel around the world a lot, and you can travel. You usually sum up a group of people. Like the French are kind of, don't really like the French, but you should all be proud of your culture. Everyone I've met has been very gracious, generous, and kind. So you should be proud of that. So today, I will be talking about fullerenes and the pharmaceutical applications of fullerene derivatives. I'm an immunologist by trade, so I work with the immune system and have lots of animal models of the immune system. So I'll start my talk with an overview of, basically the talk will be divided into three groups. We'll be talking about diagnostics using metallofullerenes. These are fullerenes that have metals inside of them, and I'll specifically be talking about fullerenes that have gadolinium. For anyone that knows anything about MRI contrast agents, when you go get an MRI, they can shoot you up with MRI contrast agents, and ours is, the trimetaspheres shown here are more sensitive than the current contrast agents on the market. I'll also be talking about empty cage fullerenes, which are very potent antioxidants. Everyone is usually familiar with the benefits of antioxidants. Fullerenes are some of the most potent antioxidants known to man. And then I'll conclude my talk with some studies we're doing in theranostics, where we actually blend both the diagnostic and the therapeutic component together. So the first leg of my talk, I'll be talking about diagnostics. This is what's called a trimetasphere, discovered by a guy named Harry Dorn at the Virginia Tech in Virginia. It's a C80 cage, has 3 gadoliniums per molecule or per fullerene, held together with a nitrogen. Now with the 3 gads, it's more sensitive than current contrast agents on the market, such as Magnavist. The contrast market is a multi-billion-dollar industry in the United States, but we think ours might be safer because gadolinium is very toxic and it's enclosed within this cage, so the body's immune system doesn't see that toxic metal. We also think it may be safer because of that, and also it can be targeted, meaning any biomarker in the body we can theoretically add to this and send it to where it wants to go. And I'll give you some examples of what we're doing with atherosclerotic plaque. So one of the disease models that we have in our lab is atherosclerosis. Everyone knows what hardening of the arteries is, when your monocytes, when you eat too many cheeseburgers or you eat too much fat, your monocytes, which is a white blood cell, get very sticky, and they stick to your arterial walls, and that's how it builds up plaque. And when that plaque ruptures, that's how you have a heart attack or a stroke. Well, we have a model of this in our lab, and one of the things that we know is that those plaque molecules have macrophages in them with what's called CD36. And so we've designed a compound with this plaque target ligand, which is actually a CD36 ligand. So the CD36 receptors on the plaque, the ligand here is on our molecule, on our trimetasphere with the gadolinium. So in theory, it should go straight to the atherosclerotic plaque. And so we made several different compounds, put them in a liposome. The trimetaspheres were intercalated in the liposomal membranes. And the model that's shown here is called an ApoE mouse. These are mice that are prone to atherosclerosis, so they actually get lesions in their blood vessels. And sometimes they get so bad that they'll have heart attacks and strokes and die. But we've used them as a model to test our plaque targeting contrast agent. So we use that trimetasphere attached to CD36 ligand and inject. This is a pre-injection looking at 3 different plaque targeting contrast agents, and looking at this is the descending aorta of a plaque mouse. So these mice have a bunch of plaque in their vessel. So this is where the blood would be flowing. So we inject our compound at 30 minutes, 60 minutes, and 120 minutes. You can see If you compare